Good afternoon, everyone. I welcome you to another series of our lecture on bacteriology. Today, we'll be looking at Enterobacter and Klebsiella, one of the two important members of Enterobacteriaceae that we'll be looking at. My name is Bamidele Odumosu. Thank you for listening. The first genus we're looking at today is Enterobacter. So Enterobacter, genus Enterobacter, is one of the important genus among Enterobacteriaceae. Enterobacter itself is an important bacteria that is well known as an opportunistic pathogen. So this bacterium was initially named Enterobacter aerogenes and was later included in the genus Enterobacter in 1960. This is based on the alignment and similarity of the sequence of other members of Enterobacter. So, Enterobacter aerogene remains one of the clinically relevant, medically relevant, environmentally relevant member of genus Enterobacter. They are facultative anaerobic rod-shaped non-spore forming bacteria, and like I said, they have emerged as an opportunistic bacteria implicated in nosocomial infection among especially intensive care patients. Enterobacter comprise of other members that are not so popular, but they also have their own significance, and they comprise of numerous species with distinct phenotypic and genotypic characteristics, and this includes Klebsiella, aerogenes, Comicola, Sakazaki, and Omache. Like I said previously, the ones that are relevant to most study include Enterobacter aerogenes. Are ubiquitous in nature. They can be found in a wide variety of environments, such as the soil, water, sewage, feces, and human guts. In the soil, they are found active, and their activity also include interaction with other microorganisms to increase the nutrient availability, and also to form part of the microflora of the soil. And also they can be found in freshwater and marine environments where they have adaptability for the presence of other inhibitory substances in the marine environment, and they also have presence in the sewage. They can be found in the sewage and feces of humans and animals. Being an enteric bacteria, they are dominant in the gut microbiome, where they also partake in the gut maintenance. They can thrive in diverse environmental conditions, and they tolerate a variety of temperatures. That is, they can grow low, and they can also grow at optimum temperature that is available for many mesophiles. So they also have adaptability to various pH levels and availability of nutrients. There are many organisms that can't survive this variation in temperature and pH level, including nutrient availability. So Enterobacter is not in the class of fastidious organism, it's not a class of a strict organism in terms of temperature and pH level. Its ability to thrive in these various conditions. It is considered as a very versatile bacteria, and this broad environmental adaptability makes Enterobacter a significant contributor to the natural microbial world. So they partake in contributing to the microbial world, making microbial world a very versatile community with microorganisms that have broad adaptability to various environmental conditions. Now, in the characteristics of Enterobacter, usually they are all the same character, they all have the same character based on member of Enterobacteriaceae, such as being gram negative and so on. But in its own shape, it is a short rod. It's not a slender rod shaped um, bacteria, just like other prominent members. Its own shape is a bit short. So these are one of the unique characteristics of Enterobacter, though it is also facultative anaerobe and motile, and is a non-spore forming and lactose fermenter. And Enterobacter usually has a close similarity to E. coli. So the the way Enterobacter and E. coli are differentiated from one another in the laboratory is by citrate utilization tests. These citrate utilization tests indicate that Enterobacter can utilize citrates as the sole carbon, while E. coli may not. Now, these tests differentiate E. coli and Enterobacter, and also the VP test is also a test that differentiates Enterobacter from E. coli. Enterobacter is positive for both tests, and they are less mucoid in terms of physical appearance. Now, Enterobacter are noted for their antibiotic resistance profile, including the fact that they are implicated in various infections. They are also useful in biodegradation and plant growth promotion. That is the environmental significance. Among part of the clinical significance is that they are implicated in urinary tract infection, bloodstream infection, and pneumonia, although they do not rank among those that are causing this infection. They do not rank top among those causing this infection, but they are among the organisms that have been implicated either in a polymicrobial infection. When you say polymicrobial infection, means microorganism like 2 or 3 causing an infection, or as a single infection in the hospital where they are usually found lurking around, forming biofilm around medical equipment, and they increasingly being implicated among multi-drug resistance to last line of antibiotics. The last line of antibiotics being carbapenem has been reported as being resistant to carbapenem. So Enterobacter ranks among resistant bacteria according to WHO in the listing of antibiotic resistant bacteria, and Enterobacter also is among the escape bacteria. Escape bacteria mean clinically relevant antimicrobial resistant bacteria that are found in the hospital. So Enterobacter is one of the clinically relevant bacteria that poses challenge to treatment of infection that are associated with them. and this leads to economic burden because the implication of their being member of the resistant bacteria leads to extended hospital stay, additional diagnosis tests, and high cost of antibacterial treatment. The Enterobacter Clausae is used in biological control in plant production. So they are also very relevant in control of disease associated with plants which are caused by microorganisms. Rather than using some chemical agents in controlling this disease, Enterobacter can also be used, and they have been reported to be effective as a biocontrol agent in plant production, and they are eco-friendly, which means they do not pose any harm to the environment. Our next organism is Klebsiella. So we will look at Klebsiella as another member of Enterobacteriaceae in this bacteriology. The genus Klebsiella consists of several species, and among these species we have Klebsiella pneumoniae, Klebsiella aerogenes, and Klebsiella oxytoca as those that are usually implicated in several cases of infection and mostly encountered in the environment. Generally, these organisms can be found in the soil and water and on some plants, and some of the strains are also considered part of the normal flora of the human gastrointestinal tract, where they also play a role in the microbiome of the human gut. The organism was initially named after Edwin Kleb, a German physician who isolated the organism and renamed the organism, which is Klebsiella. And ever since then, Klebsiella has been known prominently as a member of Enterobacteriaceae, which has been implicated in so many cases of infection. Their normal habitats are three major parts: the environment, where they are found, like every other prominent member of Enterobacteriaceae, are found in the surface water, they are found in the sewage, soil, and plants. They thrive in organic matter, and they contribute to the nutrient cycle just like other members of Enterobacteriaceae that are found in the soil. So they contribute to the nutrient cycle by also taking part in the improvement of the nutrients available to plants. And they are also found in the intestinal tract of humans and animals, where they colonize the guts and they play role in the digestion and guts health, maintaining and improving immune development. And they are also found as an important principal pathogenic reservoir for transmission of infection in the hospital. So they are a notable member of nosocomial pathogen. They are not opportunistic pathogen. They are actually nosocomial pathogen, and they are usually found adhering to medical equipment, hands of hospital personnel, and beddings, tables, and flaws of the hospital environment where they are responsible, where they are easily transmitted to immunocompromised patients as disease-causing bacteria. The biochemical characteristics, they are non-motile, urine positive. On Macconchia, Klebsiella colony appear with a large mucoid lacto-fermenting pink colonies. They have a mucoid colony as seen on this picture, and on triple sugar ion agar, they produce a yellow slant and gas bubbles. They are also VP positive. Now, these biochemical characteristics are peculiar to Klebsiella. However, one very important feature of Klebsiella is their mucoid appearance, which makes Klebsiella to be prominent among members of Enterobacteriaceae. They appear with a mucoid appearance, and this allows is also one of the adaptive features that allow them to stick to devices, to stick in a biofilm, and is also a form of their adaptive features. In the clinical significance of Enterobacter, they are responsible for bacterial pneumonia, especially in patients on ventilators or patients that have underlying medical conditions. Pneumonia can be caused by other factors, but when pneumonia is associated with bacterial infection, then Klebsiella rank first among bacteria that are implicated. An example is Klebsiella pneumoniae. They are also responsible for urinary tract infection, and such can also lead to complications such as pyelonephritis, which is a complication of urinary tract infection. It can also be involved in wound infection, which can occur after people have undergone surgery or trauma. And also sepsis. Enterobacter has been implicated in life-threatening systemic infections. So these are few among the clinical significance, though prominently known for Enterobacter. Klebsiella. Economic importance of Klebsiella includes the fact that they increase hospitalization among patients that are admitted in the hospital based on the fact that they can cause several kinds of hospital-acquired infection. Hospital-acquired infection, also known as nosocomial, are infection that occurs 2 to 3 days after a patient is admitted in the hospital, and these can lead to morbidity and death if not properly um, taken care of. And they also develop antimicrobial resistance, requiring more expensive drug and long-lasting treatment procedure because of difficulty in treatment of disease they are causing. Other members such as Planticola and strains of pneumonia has been isolated from the root of plants where they act as nitrogen-fixing bacteria and responsible for nutrient recycling. Thank you for listening. We will continue with the next microorganism which is coming soon.